I'm going to talk about a different issue. I'm going to be much less specific and much more general and a little bit more conceptual. And I'll try to keep to my 15 minutes if possible. So first of all, we need to understand that all biological organization is based on cells. So cell, I would say, is the fundamental biological unit. And cells, as you can see here, they're all different. They have all different genomes. And the genomes, as they explained, will contain information that allow us to create very different structures, like in this case, for example, this unicellular organism, or this plant cell, which has a very different structure, or this human cell, which is a sperm, which is a very, very special type of cell that has a very specific role. So if we think about fundamental biological units, we need to think about how do they survive and how do they develop. And the main theme of our team is cell division. So I wanted to just to give you an idea of what cell division is. This is a field of cells in a petri dish. So these are being grown in culture. And you can see here cells undergoing division. They are dividing all the time and they are creating new cells. And you can see that they are highly visible. This is, of course, done in a very fast time-lapse way so that we can actually see the events happening. Now, when is cell division required? Cell division is required for all types of organisms to survive. So cell division is required for unicellular organisms to maintain. And as cells divide, they become old and they generate new cells. So we always talk about the mothers and the daughter cells. And when we talk about multicellular organisms, cell division is a fundamental moment in the process. And here we see a human oocyte fertilized. You see the parental genomes, the maternal and paternal genomes here fusing. And this they will give rise to 2 cells and then 4 cells and 8 cells and so on and so forth. And cell division is required for this organism to grow and to eventually become what we have here as a human, as a late stage fetus and ready to be born human organism. But cell proliferation and cell division is not something that only happens during the development of a multicellular organism, or that happens to unicellular organisms when they divide. For example, in a multicellular organism, and here we have an example of such a multicellular organism, we have a huge number of cells. This is comprised by 10 to the 4th number of cells. And one of the amazing characteristics of this organism is that it is estimated that about 90% of these cells are actually renewed every week. So cell proliferation is something that is happening all the time, and it is what actually maintains our bodies working. We are made of about 200 different types of tissues, and each one of these tissues, except for a few cases, like in the central nervous system, all of the tissues are maintained through time by cell proliferation. So imagine that you have a human being in which every time a cell divides, you would see, let's say, a little flash of light. We will be glowing all the time because we have billions of cells dividing at any one time. But this brings us to another important consequence, which is that if we look ourselves in the mirror, for example, today, and we look at ourselves in 2 weeks' time, although we are still the same person, we still have the same physiognomy and with the same morphology, we actually change. Our body, the material that's actually making our body is changing over time, and as we survive in time, these cells are being renewed all the time. The real problem of cell proliferation is that things not always go right. So we have two main questions real here. One is as we grow old, our cells are no longer able to divide properly, and they don't give rise to cells that have the same capacity, and therefore we age. And in the other side, we have the question of disease. That is, when cells go completely wrong, they go completely wild and begin to proliferate abnormally. And in that sense, we begin to have cells that go out of the tissue. They can start proliferating in different parts of our bodies and create metastases. And therefore, we need to understand what are the bases of the cell division, how the cells are maintained with the right identity, so that the organism survives properly over time. So the main question about the cell division is we need to understand a little bit about what the cell goes through as the cell goes through what we call the cell cycle. So the main point about just going back to one slide, we need to understand why cells undergo abnormal cell division, so that then the cells that are produced are no longer carry out the same functions that they had before. And the question really is that cells make mistakes. I mean, if we have millions of cell divisions going on, some of those cells will make mistakes, and those mistakes will be essentially an abnormal distribution of their genetic material. And the idea is that they lose information, or they gain information, or they have abnormal information of the genetic material, and therefore the final identity of the cell becomes abnormal. So how did the cell actually go through this process of cell division? First of all, we need to have a look at here what is the cell cycle. Cells are born as cells in a phase which we call G1. All cells are the result of a previous cell division, and therefore they are created, and they are born in this stage. Then they go through a process by which they grow, and once they grow, they have to take a decision. And once they grow sufficiently, this decision can be taken, and this is an interesting decision because it's an irreversible decision. When the cell decides to divide, it will have to go through a process which involves the replication of the DNA. They explain what DNA is and what the genetic material is there. So the cells have to first replicate the DNA, and then they have to prepare for the final stage of their division cycle, which we call cell division or mitosis. Mitosis is a series of stages in which the cell has to reorganize completely his cytoplasm and his nucleus, and it has to create the conditions so that the two daughter cells that are formed at the end have exactly the same genomic content. Because if they don't, then things can go definitely wrong. Here you have a human cell. This is also a tissue culture cell, and you can see here the chromosomes in the middle, and you can see how the cell will separate the two complements of the genomes because it duplicates the genome and eventually forms the two cells at the end. Okay, this is a scheme that tells you a little bit about what happens during cell division. So the stages are, as the cell goes into cell division, what happens is that the two different organelles begin to form, these are called the centrosomes. These green things here. Well, the other thing that happens is that instead of having this spaghetti sort of mixture here in the nucleus, now we have these very organized chromosomes. Each chromosome has actually two copies of that are equal, and they're called sister chromatids. And these sister chromatids form a single chromosome. As the cell progresses through mitosis, these two centrosomes move to opposite sides of the cell. And what it begins to happen is they begin to generate fibers. These fibers are called microtubules. And these microtubules eventually have to interact with the specific area of the chromosome called the kinetochore, and they have to link. And as you can see here, each chromosome, each sister chromatid would have to bind to different poles of the cell. This is the only way in which the chromosomes can be separated correctly in the final stage, where the sister chromatids here separate, they begin to migrate to the different poles, and we end up having at the end two cells, which now here will have to reform the nucleus and eventually will divide, and each cell has a complete content of the genome. Now, the sort of critical phase of cell division is described in this film. This is a cell that is undergoing cell division. It has condensed its chromosomes. They are tightly condensed. They begin to align in the middle of the cell because these fibers that you don't see here, but the fibers are pushing the chromosomes to the middle of the cell. And suddenly, at a specific moment, something fantastic happens, which is out of this very complicated mess that you see in the middle, a very organized distribution of the chromosomes go to the different poles of the cell. I'll just show you this again, because I think this exemplifies the final stage in which the cell can actually control what is happening. And what the cells has to do is that each arm of these chromosomes has to bind to different poles of the cell. And by doing so, this ensures that one of the cells inherits one complete set of the genome, and the other cell will inherit the other complete set of the genomes. Now, this doesn't occur exactly like that. That was a beautiful cell doing it very beautifully at the first time. But most cells, what happens is they go like this. You know, they have one chromosome, for example, that moves to one pole, another chromosomes that move to the other pole. And only when the chromosomes actually organize in the middle and each sister chromatid binds to microtubules to the different poles, is that then the sister chromatids separate and the cells will exit mitosis. So the final slide that I have is just a conceptual slide that tells you that cells actually monitor what is going on during the cell cycle. For example, during G1, the cell is monitoring the environment, and it will decide whether there are conditions for the cell to actually undergo replication. In the second part of the cell cycle, the cell will monitor the DNA, the structure of the DNA, if it's correct structure, if it has replicated properly. And finally, the cell will monitor the organization of the spindle, which is that structure that is composed of microtubules, and the chromosome orientation. And only when the chromosomes are properly oriented, then the two sister chromatids will separate and the two different cells will be formed. So that was the normal part. And what we need to understand and we need to be aware is that this process goes wrong when we talk about, specifically when we talk about cancer. In cancer, here we have cells which have too many chromosomes, or we have cells that have abnormal formation of the matrix of this spindle matrix that gives rise to abnormal separation of the chromosomes. And either there are, and therefore one of the consequences is instead of inheriting a normal genomic complement, we have cells that a tumor cell will inherit, for example, 3 copies of chromosome 6 or 3 copies of chromosome 3 or bits of chromosomes that are abnormal. So understanding the way in which chromosomes segregate is essential to understand one of the major causes of, or one of the major observations that we have in cancer cells, which is this idea of chromosome instability. In this type of issues, in this type of diseases, what we find is abnormal distribution of chromosomes, and the abnormal distribution of chromosomes gives rise to very abnormal formation of the organism. So that was my presentation. I hope I gave you an idea of how important it is to understand cell division and how beautiful it is to view cell division in real time.